No questions over the homework? Population growth homework? Yeah.
Can we just go over the equation and like process of getting the population, like the 23?
Which, oh, that, okay. So there are a couple of different ways of going about this. So for Monday's exam, you're gonna have population growth stuff on there. You're gonna have to do some equation work if I know Dr. Judd, and I think I know Dr. Judd. So one of the mistakes that students make is they oftentimes confuse per capita growth rates versus raw births and deaths. So remember that raw births and deaths are the big B and the big D. So in your homework, how many births did you have? You had 5 births and you had
2 deaths.
2 deaths, right? And how many individuals did you have in the population that were producing these births and deaths?
20.
You had 20. So that's, n at times 0 is 20. So there's equations in your homework that show that the raw number is due to the per capita rate of birth multiplied times the population size, right? So what does per capita mean? I'm pretty sure Dr. Judd gave you kind of the literal definition of what per capita means. What does capita mean? It means head. You have a cap on your head. That's where that cap word comes from. Per head, per individual. So the per capita growth rate is kind of the growth rate averaged across all the individuals in the population. Not everybody is giving birth at the same time. Not everybody gives birth in a given year. And so it's the number of births divided by the population responsible for those births. So think about this. This is 5 individuals born out of 20 individuals multiplied times the 20 individuals that are there. This cancels out, giving you the 5 births that you have. So it's always useful to work through what the equations actually mean. In the same way, you can divide this by population size, and B sub t over the population size is the per individual birth rate. Okay, so it should all work together. So when you go to calculate the growth of the population, you could just say, well, we started out with 20 individuals, we added 5, and we took away 2, which would give us 23, right? That's easy enough. But if you're doing this generation after generation after generation, this becomes cumbersome. Because now you have 23, well, because you have more individuals, you might actually have more births and more deaths. And you don't know what that is. It's not always going to be 5 and 2, which is why we want to calculate the growth rate. What's the growth rate? We have 2 different growth rates that we need to think about, right? One of them is signified as lambda, and this growth rate is done. One of them is signified as lambda. What's the other one?
I got the spreadsheet.
Lowercase r. So what is lambda? Lambda is, so it's the finite rate of increase. That doesn't mean anything to you, does it? No, it doesn't. So what is lambda in equation land? 1 plus r. What is r in this case? It's r is little b minus little d. The little b's are the per capita rates, which I cannot erase. Right? So, if we want to know what r is, we need to take the number of births divided by the total number of individuals. That's 5 divided by 20, minus the number of deaths divided by the total number of individuals. This is 0.25 minus 0.1, right? So that would be 0.15. You guys follow how I got there? Okay. So that plus 1, 1.15, which is what lambda is. So one of the equations in your handout is that the population at time 1 is equal to the population at time 0 times lambda, right? What's the population at time 0? Lambda is 1.15. What's 1.15 times 20? 23. What's 20 plus 5 minus 2? 23. Okay? It should all check out if you do this. This is kind of self-checking sort of work. Now, if we want to know what the population at time 2 is, what do we have to do?
1.15 times 23.
1.15 times 23. Because this is lambda times population at time 1, right? This ends up being what, 26 point something? 0.45. 0.45. We want to know what the population is at time 3. What do? Lambda times the population at time 2. And I don't know what that is. It's 26.45, whatever that is, times 1.15.
29 something.
29 point what?
29 point something. I don't know what that is.
Whatever. So let's get rid of the numbers and just deal with the symbols. In the population size at time 3 is equal to lambda times the population at time 2. What's the population at time 2?
26.
No, in symbols. It's lambda times the population at time 1. What is the population at time 1?
R plus 1.
Lambda times the population at time 0. So if you want to know what the population is at time 3, it's the population at time 0 times lambda times lambda times lambda. That's where we get this equation. Then it's really easy. If we know what lambda is, we're going through 20 time steps because we want to know what n at time 20 is, and we know what the population at time 0 is. That's easy now. And now you know not just, you can memorize this equation, that's fine, but it's easier to remember the equation if you remember where the equation came from. Does that make sense?
Yeah.
Don't be afraid of math. Math is your friend. Try and understand math. As soon as you understand math, math ceases to become mysterious and hard. Okay? Don't give me that face. And you should be wearing a mask. Why do you not have a mask on that face?
I have to guess the guy.
Oh my gosh.
I know.
Checking this thing you learned it from.
Anybody have a spare mask for him? Where are our masks that are supposed to be in here? The advantage to wearing a mask is I don't have to see you grimace when I say things you disagree with. She's got one for you. Good. Awesome. Thanks. So the way to get over a fear of math is just to understand where the math is coming from, and then it doesn't have any power over you. Lambda is equal to e raised to the r. This is another equation in your list of equations, right?
Yeah.
Anybody have any idea what this means? Okay, e is 2.7818 something. It's one of these irrational numbers that just goes on forever in the decimal world. It's the base of the natural log. It's the base root of the natural log. So if you want to know what r is in this, you take the natural log of lambda, and that gives you r. It's just basic. Probably if you took calculus in high school, you learned this. So if you know what lambda is, which is 1.15, you can take the natural log of that, and that will give you what r is. So the natural log of 1.15 is 13, somebody remember? 139. 98. There's a 7 in there somewhere, right?
Well, it goes 13976.
13976. Okay. So this is the continuous or instantaneous rate of increase. So in the example, you have these tortoises, they live in the Galapagos Islands. All of a sudden, one day, 5 tortoises hatch, and at the same moment, 2 tortoises die. So, at time 0, there were 20 tortoises, and then we get to time 1, 5 tortoises hatch, 2 tortoises die, and we're now at 23. Stays at 23 for a while until time 2 comes along, and then, oh, I erased the numbers, it's 26.9, right? So 26, so let's say 5 more tortoises are hatched and 2 die, we're now up to 26. This is what it means when we talk about a finite growth rate. Finite growth rates are calculated for these organisms that live and die in these pulses. Did you guys hear about brood 10 this summer? The cicadas? You guys didn't hear about the cicadas? Cicadas, these periodical cicadas occur in these different broods, and different broods emerge at different years. And this year in the northeastern United States, brood 10 was emerging in the northeastern United States. And this is a species that hangs around for 13 or 17 years, depending on the species. And every 13 or 17 years, all the cicadas come out of the ground as larvae, as nymphs. They transform into adults, they sing a lot, they make a lot of noise. People take pictures of them because they're everywhere. And then they breed, they lay their eggs, and they die. And then those eggs hatch out, and then they go underground and they feed on trees for another 13 to 17 years. And then they all come out, they transform into adults, they breed, they lay eggs, and they die. Those kinds of species can be modeled with lambda because the death and birth isn't continuous. It happens in these discrete spurts. This is sometimes referred to as the discrete rate of increase also. But that's the kind of reproduction and death that is being modeled. Are humans like this?
No.
No, you can give birth and die at any moment. Just ask anybody who's giving birth right now, and ask all the people who are currently dying in the United States. It can happen at any particular time. In this case, the growth doesn't look like this stepped kind of curve. It looks like a continuous curve because at any given moment, individuals are blinking out and individuals are being born. Because that happens at any given moment, the overall value of this is way lower because this gets multiplied over little tiny time steps, infinitely tiny time steps. And so that's why the value, the magnitude of this is so much lower than the magnitude of lambda, because this is an instantaneous. What is the growth rate in a population at any given small interval of time? Well, in your list of equations, there's an equation. If you want to know what the population is at time t, all you need is the population at time 0 multiplied times e to the rt. R is this instantaneous growth rate. T is the number of time steps. So if we want to know the population at time 1, then all we need to know is what is the population at time 0 times e to the r times the number of time steps, right? You guys do the math on this? What does it come out to be? No, just one time step, not 20 time steps. Yeah.
23.
When I did it, I got 22.999999999, but that's basically once again, these equations are self-reinforcing. We should be able to approach this from a variety of different perspectives, and it should still give you roughly the same answer, or in this case, very, very close to the same answer, if not exactly the same answer. So, when you're doing these kinds of things, try and get to the reasons why the equations are doing the things that they're doing. That makes the equations easier to approach, and that's the way you're going to succeed. You can just try memorizing the equations, in your homework, Dr. Jed put in there that the equations are not gonna be on the exam. We put the equations on the exam every year. Guess what? If you don't know how to do it, the equations don't give you a damn bit of help. Learn the how, and then you don't really have to rely upon the equations. And if you learn the how, when you look at the equation will actually make sense to you. Questions? Is this clearer now? Okay, all right, good. I have papers to hand back to you from Dr. Judd. He was very busy grading yesterday, getting your population homework graded. Population homework that you did as practice homework for today, keep that so that you have something to study for the exam on Monday. The practice homework, yep, just keep that, don't turn it in. I was helping a couple of students out earlier today. I went basically kind of what I just went through with you, and when I got to the end, and 23 was in one place and 23 was in another place, they were like, oh, I'm like, yeah, see, that's why we do homework. Some of you think probably that the reason that you're doing homework is because when you turn the homework in, you get points for that homework. You know how many points that homework adds up to over the course of the semester? We scale it, however many points it is, we scale it down to 75 points. Do you recall how many points there are in this class approximately? 1,390. That's 75 points. Is that, what was that, Charles?
I said 1309, but I guess I think so.
Close enough. It's almost 1,400 points. That's 75 points for doing homework on a daily basis. It's not going to change anything by the time of day.
Don't say that. Don't say it.
Except that in the process of doing the homework, you achieve insight into the things that we want you to know. The reason that we give you homework is not to boost your grades a little bit, which it does boost your grades a little bit if you're doing it every time. The point of homework is it helps you learn. That's the reason we give it to you. The only reason we give you points for it, because that's the only way you'll do it.
You're right.
That's true. It's true. I mean, I'm making assumptions about you, but that's probably the only way to do it. There may be one or two of you who are super motivated and you'll do it simply because it's the right thing to do. We give you this little incentive, tiny incentive to get you to do it, because in the process of doing it, you come into class and you have questions about it, or you come to my office hours and you get clarity on it. You can listen to us yammer all day long and you won't learn anything. You will only learn when you do the things that we're teaching you to learn. So, at the beginning of the semester, I had a conversation with Dr. Judd. And I said, let's not take up any homework and let's not grade it. We'll just assign the homework, and after a couple of days, we'll post a piece of homework on the Moodle site. Because what I really want students to know is that the purpose of homework is not to boost your grade. The purpose of homework is to learn. And then he made the cogent argument, if we don't give them points for it, will they do it? I'm like, well, you're super depressing and cynical. And
He's right. He's real about it.
so I saw the wisdom of his argument, which is why we're still giving you credit for it. But recognize that 75 points out of 1,390 points is really a pretty small portion of your total grade. The reason we want you to be doing the homework is because it helps you learn. And it would help you learn whether we took it up or not, as long as you were doing it. So as you approach homework, approach it with that particular type of mindset. The reason we have due dates for homework is because we want you to get it done before too much time has passed between we cover it in lecture, so you haven't forgotten all about it between when you actually do the homework and when we actually cover it in class. We've thought this through, we've debated this a lot. Do the homework, hopefully it's helping you learn the things we want you to learn. All right. Any other questions about population growth? Okay. We are slowly working through the theory of evolution via natural selection. Who can tell me what the theory of evolution, the MF question, says in terms of its 4 preconditions for it to work. Yeah.
There has to be variation.
Okay, so there has to be variation. This is my terminology. It'll be slightly different from Dr. Judge, but it's essentially the same in terms of meaning.
There has to be an overproduction of offspring so that like not, because not all of them will survive and then to make it to be able to reproduce.
Yeah.
There has to be different fitness consequences.
So phenotypic variation leads to variation in fitness or variation in one's ability to survive and or reproduce.
It has to be heritable.
Okay. The homework you did for today and Wednesday's lecture was about this. It shows you that if populations don't have anything checking their population growth, they tend to grow in an exponential pattern. You go out 20 generations and you have, what was it? 324 or whatever tortoises from these 20 tortoises that started out just in 20 years into the future. So that's how you get more offspring being produced than could possibly survive, is because populations generally will tend to grow exponentially unless there's something reining them in. We'll talk in the second test material about the kinds of things that rein in population growth. The second one of these, this idea that phenotypic variation exists, this comes from a thing called the central dogma, which you probably got in high school. It's the story of essentially how you get from DNA to a protein through the process of protein synthesis. DNA is used as the template for creating messenger RNA. Messenger RNA goes out into the cytoplasm, it interacts with ribosomes and transfer RNA, and that process leads to the formation of proteins by stringing together these long sequences of amino acids. Mutation then brings about changes in the genetic code. Some of those changes in the genetic code lead to changes in protein structure, and that change in protein structure is what then leads to variation. And so that's what today is about. After the test, we're going to talk about genetics and heritability, and then we're going to talk about fitness. So we're kind of stepping you through slowly through each of the different preconditions. Today is about the second precondition, so you know where we're headed. So, you should be able to explain the process of DNA synthesis, or protein synthesis from DNA to protein and the various steps in between. You will receive this again when you take cell molecular biology. You will receive this again when you take genetics. This is a big, important concept. If we have time, I will also go through why you need to know this because we're in a global pandemic, but we might not have time for that. Be able to explain the different types of mutations and how they affect or don't affect protein structure, and then be able to relate protein synthesis and mutation to phenotypic variation, which is kind of the main point over there on the board. So, you guys remember DNA, right? What can you tell me about DNA? You've already said something, You said something, did I?
I haven't, but
Oh, Trevor.
it's a double helix structure.
Okay, so it's a double-stranded structure, and when it's not in this kind of fake form, it actually twists around on itself. So it's a double helix. Watson, why can't I remember Watson's first name? James Watson. James Watson wrote a book about the discovery of DNA, and he called it the double helix. So it's this helical structure, and it has these two strands. What else do you know about DNA?
cells to reproduce?
Okay, so it has information content. That information content is stored how? What is the information content? It's made up of a phosphate sugar backbone and nitrogenous bases. It's made up of nucleotides, so it's a polymer. It's a large molecule that is made up of small numbers of repeating units. And the repeating unit is the nucleotide, which is a phosphate molecule bonded to a sugar molecule bonded to a nitrogenous base. But what part of this nucleotide is responsible for the information content of DNA? Caden, you said something about information. What about information?
It keeps the structures of our cells and how they reproduce, and adenine, thymine, glutamine, cytosine. It's the pattern of it.
So it's the sequence of those nitrogenous bases which is responsible for the information content. So we have 4 nitrogenous bases. Adenine and guanine are purines. Thymine and cytosine are pyrimidines. Purines are single ring structures. So these are purines. Pyrimidines, purines are double ring structures. And purines, give me a minute. Purine, 2 rings. Pyrimidine, 1 ring. Sometimes I have to write in order to get my brain to function correctly. If you have trouble remembering this, remember that the smaller name goes with the bigger molecule. Purines are double ring structures, pyrimidines are single ring structures. So these are pyrimidines, these are purines. 2 purines, 2 pyrimidines. Doesn't really matter for this class that you memorize which are which, but just so you know that's how they are. Adenine always pairs with thymine, so whenever you see a purine, it's always paired with a pyrimidine. This purine always pairs with this pyrimidine. The functional reason that works is because when you have a single ring bonded to a double ring, this diameter of the molecule is the same. Single to double, diameter of the molecule is the same. If you get a mutation that puts a single ringed molecule with a single ring molecule, that DNA molecule sucks its waste in. If you have double rings next to one another, it makes it be fatter. And the diameter of DNA is constant across the length of it because you always have purines bonding with pyrimidines. And cells have checks that it does on DNA when it makes new copies of DNA. And one of the things that it checks is it checks the diameter. And if there's a problem with the diameter, it either gets rid of that DNA or it tries to repair that DNA. So, but the information content that cells use to make proteins is found in the sequences of these nitrogenous bases. And that's one of the important things. So I came over here to give Caden a point. I need to give Trevor a point. Trevor, you have your own pen. And then Trevor, did I remember you were going to say something?
Sadie?
Oh, yeah. So you get 2 points. Oh, it's the other one. Oh, sorry.
Put 30, that's a lie.
All right. So that's the basic structure of DNA. And that's how information content is stored in DNA. Forget this. These 2 strands of DNA are held together through hydrogen bonds. If you don't remember hydrogen bonds from high school, you can come and talk to Dr. Judd and I. These are weak intermolecular forces. One of the nice things about them being weak is that they're easy to break. And so I would normally go through and tell you why hydrogen bonds form, talking about electronegativity, but I'm going to not do that because we're a little pressed for time. But those hydrogen bonds, because they break easily, it opens up DNA so that so that when you open up DNA, you expose these nitrogenous bases, and you can add new nitrogenous bases to it. So when you need to make a new copy of DNA, it's easy to break DNA apart and then add new nitrogenous bases and the nucleotides that they're attached to the open strand of DNA. So here we have a double helix of DNA. Here we have the purple strands are the old DNA. The pink strands are new DNA where the nucleotides have been added to the open nitrogenous, open unbonded nitrogenous bases. And it's just basically adding an adenine to a thymine, thymine to an adenine, guanine to cytosine, et cetera, et cetera. From this single DNA molecule, you can then build two new DNA molecules, but each of those new DNA molecules is made up of half of it is old DNA and half of it is a new strand of DNA. Because of this, we call it semi-conservative replication, because half of the new molecule is made up of old template DNA and the other half is made up of newly constructed DNA. This is the process of DNA replication. This is what cells do before they're getting ready to go through mitosis or meiosis. In order to divide the DNA into two new cells, you have to have enough of that DNA to go into those new cells, so you have to duplicate your DNA. That process is called replication. We're not going to spend a ton of time on that in this class. Once again, you'll get a lot more about the mechanics of that in genetics when you take genetics at the end of your second year. In the same way, you can use DNA to create messenger RNA. Similar thing happens, except that rather than opening up the DNA molecule totally, you open up a little section of it. It's called a replication bubble, because it looks kind of like a bubble when you actually see it in real life. And then there is an enzyme called RNA polymerase. The enzyme that does this for making DNA is called DNA polymerase. There's an enzyme called RNA polymerase, and what it does is it creates a new strand of genetic material, but this is messenger RNA rather than DNA. And it creates a single strand, and when it's finished making that messenger RNA, the messenger RNA gets broken off and it gets transported to the cytoplasm. Now, who can tell me the differences between DNA and RNA?
In RNA, the thymine is placed with uracil.
Okay, how else do they differ? Somebody else?
RNA is a single strand.
RNA is single strand. Any other differences that we need to be aware of? RNA is used to make proteins, but then technically so is DNA, in that DNA specifies RNA. So, you go back far enough, DNA is also involved. That's not the best, it's not a wrong answer, but it's not the answer I was looking for. Yeah. No, so DNA is used to specify the sequence of nitrogenous bases in RNA, and then RNA specifies the sequence of amino acids in proteins. Yeah.
Doesn't RNA use sugar ribose, and then DNA uses the deoxyribose?
Yep.
Is that right?
Yep. Now we'll give your group a point. Remember your name?
Clara.
Clara. Give your group a point. Oh man, you guys are racking it up, and Gaten again.
Two?
Yeah, two, because it was gonna work and Clara.
Do we get a point?
I don't know, did you say something? Who? Oh yeah, you get a point also. Yeah, call me on it, because I sometimes lose track. The most important part of this is that uracil replaces thymine. That's an important thing because you can easily recognize RNA from DNA. the structure of ribose versus deoxyribose, that's a really subtle distinction. And the other distinction that is important is that it's a single-stranded molecule in RNA, whereas it's a double-stranded molecule in DNA. And that single strand has functional consequences when you go to make a protein. So once again, you want to be paying attention to the differences that are important rather than differences that are not particularly important. I skipped over a couple of slides because this is a section that Dr. Judd normally teaches, and he didn't leave me any PowerPoint. So there's some things in here about the structure of the genome. Don't worry about those won't be on the test. It's something that I'll use when we talk about genetics. But it's in here because it's just kind of, this ultimately is going to lead to genetics, and so that's why it's in here. But I had to skip over that stuff because it's not really relevant to today's discussion. So DNA opens up with an enzyme called helicase, which breaks open the two strands of DNA. RNA polymerase adds RNA nucleotides to the strand of DNA, and when it's done, those RNA nucleotides become dissociated from the DNA, and the DNA comes back together and complementary base pairs with itself. So in this case, you create messenger RNA without ever doing anything to the original structure of the DNA. You unzip it, you use it as a template, and then you zip it back up. Now, protein synthesis then proceeds in the cytoplasm. So you make RNA in the nucleus, and then it's shipped out of the nucleus into the cytoplasm, and the cytoplasm are where ribosomes occur. And ribosomes occur as these 2 units of proteins and RNA. So they're made up of ribosomal RNA. So there are 3 types of RNA that you need to be aware of. There is mRNA, which is messenger RNA. It's called messenger RNA because it carries the message from DNA out into the cytoplasm. I already mentioned that there is ribosomal RNA. This is RNA that is used in the construction of ribosomes. So that ribosome is actually a complex of protein and RNA. What's another type of RNA that you're aware of? Yeah. This is transfer RNA. Why is it called transfer RNA? Someone else. What group are you in? Okay, so it's a non-omic. I knew that would happen. What's transfer RNA about? Why is it called transfer RNA?
You transfer the proteins.
Transfers amino acids to the ribosome, so it's like a carrier molecule. You guys are.
Yeah.
What group are you in? Oh, we're here.
Yeah, right there.
So transfer RNA essentially moves amino acids to the ribosome. Yeah. All 3 of these RNA molecules are specified by sections of DNA, by genes in DNA. What is your guys' carrying around definition of what a gene is? If your parents said, well, what are you learning in biology? And you said, well, we learned what a gene is. Right now, what would your definition of a gene be? Okay, so a trait, it's not really a trait because the trait is what you see. It's what Dr. Judd has been talking about when he talked about phenotype. So like eye color would be a trait. Specific phenotypes within eye color are gray eyes and blue eyes and green eyes and brown eyes, et cetera, et cetera. Those are different variations of this single trait. But what causes those variations? Yeah, so the genotype, so there is a gene somewhere, there are actually multiple genes that control eye color, and those genes are what give you the specific phenotype for the trait eye color. But what is a gene then? Okay, so it is genetic, so refine that a little bit. When we're talking about genes, we're talking about DNA, we're talking about RNA, we're talking about proteins. Okay, so it's a sequence of DNA. Remember, a nucleotide is a phosphate bonded to a sugar bonded to a nitrogen base. So it's a sequence of DNA nucleotides, and what do these nucleotides do? Yeah.
Code for trait.
They code for a trait. Okay, so they code for a trait. This is a classic high school level kind of definition for what a gene is. It's the kind of definition that I learned when I was in high school. Guess what? You're not in high school anymore, you're in college now. Are these traits? No, they're just molecules, aren't they? So we used to teach students, at least when I was in high school, we would teach students that a gene is a sequence of DNA nucleotides that code for a particular trait or code for a particular protein. We were even more specific about it. Codes for a protein. And then they would teach us about protein synthesis, and then we learned that messenger RNA is actually formed by genes found in DNA. So how can we refine this definition to make it a little more realistic? Because it does code for proteins ultimately, but it also codes for messenger RNA. Yeah. Okay, so does it code for things other than messenger RNA and proteins? You're going to learn about all kinds of different things that genes code for when you get to genetics. They're what they call extrachromosomal DNA that is floating around in the nucleus and in the cytoplasm. There are all kinds of products of genes that you're just not aware of yet because you haven't yet gotten to genetics. So the definition that you will probably get in genetics is that sequence of DNA nucleotides that code, that code for a functional product. And that functional product could be a protein, but that functional product could be messenger RNA, transfer RNA, ribosomal RNA, microRNAs, all kinds of different products that come out of DNA with DNA being used as the template for creating those things. And they're actually just too numerous to really mention in any sort of way in a sensible walking around definition. But the point of this is that it codes for something that actually does something, has a function. And in this case, the things that we're mainly concerned with are these different types of RNA molecules. So DNA has genes that specify proteins, and those sequences of DNA will be transcribed into messenger RNA. You guys know what I mean by the term transcription? What do I mean by transcription? Yeah. Transcription is the process by which DNA is opened up. You build messenger RNA nucleotides onto that template, forms a messenger RNA strand, and you've basically taken the information content of DNA and you've transcribed it into information content in messenger RNA. So transcription. Transcription is also the process of building new DNA from old DNA. It's just one is the transcription of RNA, one is the transcription of DNA. So the messenger RNA is transcribed from a gene in DNA that is actually specifying the code for a protein. Because when that messenger RNA gets out into the cytoplasm, it hooks up with the ribosome clamps around it, and then the ribosome interacts with transfer RNAs. So transfer RNAs are floating around in the cytoplasm. It's an RNA molecule that is attached to an amino acid. And RNA generally doesn't form a double-stranded molecule, but it does in transfer RNA. So in transfer RNA, you have a bunch of complementary base pairing. Then you have regions where complementary base pairing doesn't occur, and you just have nitrogenous bases hanging off free. Then you have other regions that are paired, but then you have regions where the nitrogenous bases are just hanging off. And you have another region where they're paired, and then you have another region where they're just hanging off. And then at this point, attached to these free nitrogenous bases, there's an amino acid bonded to that. Down here, there's a sequence of 3 nitrogenous bases that are important. This is called the anticodon. And so the anticodon is made up of nucleotides that have exposed nitrogenous bases, and for whatever reason, the resolution on this projector is terrible. And in this case, this first anticodon at the top of here is UAC, uracil adenine cytosine, and it's the anticodon at the end of a transfer RNA molecule that has a methionine amino acid attached to it. Those nitrogenous bases are just like any other nitrogenous base. They want to complementary base pair with other nitrogenous bases, and this is going to base pair with an adenine, a uracil, because we're talking about RNA now, and a guanine. So you see AUG and you see UAC. I mean, you can't see UAC because it's very blurry. So what this transfer RNA molecule does is it complementary base pairs with the sequence in messenger RNA. In the process of doing that, it brings its attached amino acid to the ribosome, and it puts it in this first active site. There's an active site here, and there's an active site here. There are other transfer RNA molecules out there with other anticodons, and they're carrying other kinds of amino acids. In this case, this looks like this is a CUG, so this would be a GAC anticodon, and this one happens to carry the amino acid leucine. Once that first transfer RNA molecule is in there, in the first position, a second transfer RNA molecule will come in there and occupy the second position. Once you have 2 transfer RNA molecules sitting there next to one another, these 2 amino acids are right next to one another. One of the functions of the ribosome, the primary function of the ribosome, is to get these 2 transfer RNA molecules close to one another, and then it has an enzymatic function that basically forms a bond between the first amino acid and the second amino acid. A peptide bond is the kind of bond that forms between 2 amino acids. Once that peptide bond forms, the bond of methionine to this first transfer RNA molecule breaks, and the ribosome kicks that transfer RNA molecule out. As soon as that transfer RNA molecule is gone, this first active site is open, and the ribosome shifts down 1 codon. This transfer RNA molecule comes to reside in the first site, which then opens up the second site for a new transfer RNA molecule to come in there. When that transfer RNA molecule comes in there, it has an amino acid attached to it. A peptide bond will form. When that peptide bond forms, the transfer RNA molecule in the first site gets kicked out. The ribosome shifts down and is ready to receive another transfer RNA molecule. And in this way, you go from a couple of amino acids to a long sequence of amino acids. How do you stop protein synthesis? There are a series of stop codons. So we know what these codons mean. So this is a codon table. You have the first base, the second base, and the third base. So if you have 3 uracils, uracil, uracil, uracil codes for phenylalanine. Oh, this is wrong. This is the first base, this is the second base, and that's the third base. I've never really looked at this that carefully, because this is A, C, and U. Adenine, cytosine, uracil codes for threonine. A couple of things you should notice about this table are what? What becomes clear to you when you look at this table?
combinations of things code for the same thing.
Different combinations code for the same thing. This is what we refer to as redundancy in the genetic code. And one of the things you'll notice about this redundancy is it usually occurs such that as long as you get the first two nitrogenous bases in a codon correctly, it doesn't matter what the third one is. So the third position is generally more variable than the first two positions. Other things that you'll notice is that there's always a start codon, but there's only one, and there are 3 stop codons. These 3 stop codons correspond to transfer RNA molecules that lack something. What do you suspect that they lack? Transfer RNA molecules have an anticodon that matches up with that stop codon, but what do they not have that might stop protein synthesis? What do you need to continue protein synthesis? You need amino acids attached to those transfer RNA molecules. Who was that? Yeah, Emma, good. These 3 codons correspond to transfer RNA molecules that don't have an amino acid associated with them. So when the cellular machinery that is adding amino acids, the ribosome, is adding amino acids, gets to this transfer RNA that lacks an amino acid attached to it, it doesn't know what to do with it, and it stops the process of creating a protein. And so it will just continue until you get to one of these stop codons that correspond to a transfer RNA molecule that rather than having a cysteine attached to it, there's just nothing there. There's nothing to form a peptide bond with. It loses its enzymatic function. The ribosome pieces become dissociated with one another. They release the messenger RNA molecule, and they stop the process of coding this long protein. Question, that is the central dogma. The central dogma is this process of going from DNA to messenger RNA, shipping that messenger RNA out into the cytoplasm, using the cellular machinery of the ribosome and the transfer RNA molecules to basically build a long sequence of amino acids that is the protein. Does this sound familiar, like something you learned about in high school? For the purposes of this class, that's all the detail that you need to know about this process, simply because it's not a genetics class or a cell molecular biology class. Questions about this? All right. So mutations, what is a mutation? If you had to give me a definition of a mutation, don't scroll forward on the slide so that you see the definition of a mutation. Yeah, Emma.
Like when something like doubles itself or it lacks something.
Okay, so those are specific examples, but what are you changing?
Chromosomes.
You're not changing the chromosome, you're changing the DNA sequence, the sequence of nitrogenous bases in the DNA. Are these changes directed or are they random? They're random. So you can think of a mutation as a random permanent change in the nucleotide sequence of an organism's genome or its extrachromosomal DNA or RNA. What do we mean by extrachromosomal DNA and RNA? But if it's extrachromosomal, what is it? It's not actually attached to a chromosome. So messenger RNA and transfer RNA and ribosomal RNA aren't bound up in chromosomes. When you think about chloroplasts and mitochondria, is their DNA the same as the DNA in the nucleus of the cell, or is it different? DNA in chloroplasts and mitochondria. Same as the DNA in the nucleus of the cell, or different from the DNA in the nucleus of the cell? Somebody mutter
Is it different?
It's different, yeah, because presumably way back in evolutionary time, these eukaryotic cells absorbed prokaryotic cells and they started a symbiotic relationship. And so the DNA in chloroplasts in plants and in mitochondria in all organisms is actually more similar to bacterial DNA than it is to eukaryotic DNA because of what's known now as the endosymbiotic theory, this idea that these organelles that have their own independent DNA were actually single-celled bacteria that were absorbed by eukaryotes and then over time became permanently associated with eukaryotic cells. So other extrachromosomal DNA and RNA, part of that is actually the DNA and RNA in chloroplasts and mitochondria. Those nuclear materials can mutate the same way that your genomic DNA can in your nucleus. So there's lots of different forms of DNA and RNA to think about. These take a variety of forms. Substitutions involve just switching out one nitrogenous base for another nitrogenous base. Deletions are losing a nitrogenous base altogether. Duplications are creating copies of little strands of DNA. And then transposable elements are things that were discovered in corn originally, where entire sections of DNA break off and then become reattached at another section of the chromosome. All of these things lead to genetic variation. Mutations, we generally think of a generally walking around idea of the rate of mutation, about 1 in a million replications. So if you have a chromosome that is 1 million nucleotides long, when you replicate it, you can expect one mutation to occur in that replication event. But they vary a lot. They vary by taxa. Different organisms mutate at different rates. And even different sections of the genome within an organism can mutate at different rates. So there is some variation in mutation rate, but a good walking around number is about 1 in a million nucleotides per replication will mutate. If you have 3 billion nucleotides that you're replicating, then that leaves a lot of possibility for a lot of genetic change in a single replication event because you're talking about so many nucleotides. So substitutions are sometimes referred to as results in single nucleotide polymorphisms. This is just when you separate a purine for a purine or a pyrimidine for a pyrimidine. These are transitions. Transitions happen more often than transversions because you're trading out a purine for a purine or a pyrimidine for a pyrimidine, so you're not changing the diameter of the DNA molecule, and that mutation goes relatively unnoticed by the DNA checking machinery of the cell. These occur at a faster rate than transversions because transversion is when you replace a purine with a pyrimidine or when you replace a pyrimidine with a purine. So you're taking a single ring structure and putting a double ring structure in there in its place, or vice versa, and that either makes the DNA molecule wider than it would normally be. Whoa, what happened? I should have had a figure in there. Where did my figure go? It either makes the molecule wider because you now put 2 double rings in here, or it makes it narrower because you put 2 single ring structures in here. And that's relatively easy for the cell to detect, and it either destroys that DNA or it goes about repairing that DNA. So if I'm recalling correct, transitions make up about 2/3 of the mutations that we see that are single nucleotide mutations, and transversions only make up about 1/3 of the types of single nucleotide mutations. Additions and deletions can have catastrophic effects on DNA because of what we call frame shifting. So if you have a DNA template, and from this DNA template, you're building your messenger RNA molecule off of this messenger RNA. AUG is the start codon, which corresponds to methionine. AAG corresponds to lysine. UUU corresponds to phenylalanine. GGC corresponds to glycine. And UAA is one of the stop codons. So this gene would code for the lamest of all proteins with only 4 amino acids before it stops creating this protein. If we start with that same DNA code though, and we insert an extra adenine in here, what it does is it inserts an extra uracil in here, and it causes all those other things to shift down. And because these codons are 3 nitrogenous bases long, it changes the codon sequence, what groups of 3 are added. And it converts what did code for a lysine amino acid, now is coding for a stop codon. So now you get an even lamer protein because it only has 1 amino acid in it. In the same way, if you add or delete, it may not have any effect on the progress of protein synthesis, but if you either insert it or delete it, the first amino acids are the same as the original code before the mutation, but all of the amino acids after that will be different amino acids. It leads to a different sequence of amino acids, and that will oftentimes lead to a protein that is a protein, but it doesn't have any function because it doesn't have the correct building blocks to do whatever that gene was originally supposed to be coding a protein to do. These are referred to as missense mutations because they create a protein, it's just not a protein that functions. And so these deletions and insertions tend to shift the codon structure, and as a result, they generally destroy the functionality of that gene. So the central dogma basically is this process of creating messenger RNA from DNA through the process of transcription. The process of using that messenger RNA to build a protein is referred to as translation. Transcription and translation are just analogies that we use. I mean, these are the terms that we use to describe them. But when you think about transcription, if you're a scribe in a monastery and you're doing this job of transcription, you basically have a book sitting on your desk beside you, and you have a book that is just an open book with a bunch of blank pages in it, and your job is to transcribe the content of one book into another book. When you do that, you may make deliberate changes to that book. You may embellish the weird figures that are around the text, but you also make a lot of inadvertent mistakes as well, misspellings and things like that. Those misspellings are analogous to mutations. When you go from messenger RNA to protein, though, you're no longer in the same language of nucleic acids, of nitrogenous bases. So transcription takes DNA, which is in the language of nitrogenous bases, and converts it into another copy of something that is in that same language, this language of nucleic acids and nitrogenous base sequences. But when you move from RNA to protein out in the cytoplasm of the cell, you're basically taking information that is essentially in one language and you're converting it into information that is in another language, going from the language of DNA and RNA into the language of amino acid sequences, and that's why you use the term translation to describe this. Mutations are just copying errors. They are random, they are not directed. It's not the scribe sitting there and adding embellishment to the illustrations, it's the scribe misspelling the word. Questions? All right, we are virtually out of time. On your way out, this was originally designed to be an in-class activity. We ran out of time. Take this home. It allows you to practice the process of moving from DNA to messenger RNA and messenger RNA to a protein sequence. Don't need that either. So pick this up on your way out, and then I have things to hand back to you. So.
Yeah.
And I don't know how Nathan has arranged these. Jamie Barkley. Sadie. Emma. Will, Courtney. Laurel, Coleman. standing up now. Moving around. Sydney, here. Mia.
Yeah.
Clara. Sarah. David.
Yeah.
Right here. Katie, Hilliard. Really? I can. Zach. Good. Hey. Savannah. Okay, thanks. Eric. Maggie Shockley.
35 double.
Maggie.
Also, you gotta try to take an outside step and step inside step. That's
I know,
your kid will just
Yeah,
not a lot.
give me a chance to get back there. Lorna. There you
I don't understand.
I understand.
go. Amanda.
If they have papers,
Light.
I already got your papers. I don't think I have to go vote. I can stay.
And then we can meet at
Hey, Lily. Why are you yelling at me?
They already walked out.
Oh, I already left. No, if you want to organize something,
Yeah.
pick a time. I mean, it would have to be on the weekend because it's not going to really do you any good.
Oh, weren't we supposed to pick something up?
Yeah,
you pick up that.
Oh, good. I thought that was all Sam. Oh, because you were Sam's, cleaning Sam's paper, sorry. Yeah, there you go. Sorry about that. Yeah, so email us. Dr. Detta's out of town for a family issue.
Yeah.
So, but I can be around. I know everything he taught on, so yeah, just be self-organizing.
Okay. That's good.